Chandan, let's start. Bhargavi will join.
Yes, madam.
Right? Yes. The most important thing in illness is never to lose hope. Quoted by Nikhil Lenin. Very good morning to one and all present over here. Before starting the webinar, I request everyone to remain muted, both your audio and video throughout the session. We have our resource person, Dr. Mathivanan Jyoti, with us, and I request everyone to maintain decorum throughout the webinar. If you have any questions to ask, kindly write it down in the chat box. At the end of the webinar, questions will be read out to the speaker. On behalf of Department of Genetics, Indian Academy Degree College Autonomous, I cordially welcome all the participants to the webinar on targeting tissue-specific transcription factors, a promising cancer treatment. Now, I take this opportunity to welcome our Honorable Chairman, Sir, Secretary, Ma'am, Principal, Sir, Vice Principal, Academic Director, who are the motivational force in conducting such events. I am glad to welcome Dr. Mathivanan Jyoti, Speaker of the Day. Welcome, sir. I also welcome heads of various departments, faculty members of various departments, and dear students. Cancer is one of the leading causes of death worldwide and poses a severe threat to society. Recent scientific advancements in cancer diagnosis and treatments improved patient survival. However, definite cure is not yet available. Further, conventional cancer therapies are often non-specific and cause severe side effects. Targeted therapy is an emerging tool in cancer therapeutics that might possess less side effects to the patients. This talk focuses on how tissue-specific transcription factors will be promising candidates to develop cancer-specific therapeutics. I am glad to welcome and introduce Dr. Mathivanan Jyoti to the gathering. Dr. Mathivanan Jyoti, Associate Professor from Department of Genetics, National Institute of Mental Health and Neurosciences, popularly known as NIMHANS, Bangalore. Sir has obtained his bachelor degree in biochemistry from Kongunadu College of Arts and Science, Coimbatore. And he obtained his post-graduation in molecular biology from Department of Biochemistry and Molecular Biology, University of Madras. Then Sir pursued his PhD in the year 2007 from NIMHANS, then moved for postdoctoral fellowship in abroad. He got trained in research at world-renowned hospitals and research institutes, includes Cleveland Clinic, Roswell Park Cancer Institute, USA, and San Raffaele Hospital, Italy. After his 7 years of postdoctoral fellowship, he started his independent research and teaching career at the Department of Biotechnology, Bharatiya University, from prestigious UGC faculty recharge program. Then he moved to NIMHANS in the year 2017 and established Laboratory of Molecular Therapeutics. His research primarily focused on normal cellular differentiation and its deregulation associated with the disease development. Specifically, understanding the genetic epigenetic regulation in the establishment of differentiation program and the development of novel therapeutic strategies for cancer, degenerative and developmental diseases. Sir has published several papers in peer-reviewed and high-impact factor journals, and his research program is funded by DST, Sir, and NIMHANS. Sir received various national and international awards to his credit. It's our pleasure to have Dr. Mathivanan amidst us. Now I request Sir to deliver talk on targeting tissue-specific transcription factors, a promising cancer treatment. Welcome, sir. Over to you.
Thank you, madam. Thank you for the elaborate introduction.
Thank you.
And before starting that, I will thank for the invitation, and I would like to thank your administration, your college administration, for giving the opportunity. So I will go for the
Thank you, sir.
talk. And today my talk is the targeting tissue-specific transcription factors, which is a promising cancer treatment. So this is the overview of my talk today. First, I am going to give little bit introduction about the gene expression and the disease development. Then the introduction about the cancer and the development of PAX3 targeted therapeutics in our lab for the neuroblastoma, and another transcription factor, Miphail, that can be a potential target in the other muscle cancer, Rhabdomyosarcoma, and some our future research or other research focus, and I will be discussing. Before going to that, I am starting from the very basic. Everyone knows, because you are a genetic student, so everyone knows that genetic information stored in the DNA. Okay, so that means the DNA has all the information to create the entire animal. Okay, approximately 20,000 genes are this approximate number. The number is going a little bit up and down also. Approximately 20,000 genes are present in our genome. And if you compare the DNA content in any individual, for me and you, it's a 99.9% are identical. Whatever the difference you are seeing, it's a 0.1% that is responsible for the variation, whatever we are seeing. And also the disease susceptibility. I will get some disease, somebody will not get the disease. This is due to the 0.1% difference in the DNA. Okay. So if you come to the gene expression, you can see the molecular dogma. Okay. Molecular biology. It's a central dogma. You maybe already studied. Anyway, I will just go briefly. So the information, the DNA will copy itself. That's a replication. Then the message which stores in the DNA, that is used to synthesize the RNA that processes the transcription, then translation into the particular protein. The protein undergoes some modification, like a post-translation modification, then its function. So why I'm saying this one, presence of the DNA doesn't mean its function. Okay. So the DNA has to make the RNA, and the RNA has to make the protein. Although proteins are present, still it's not functional. It has to modify in certain way. Then it has to go to the site of the action, whether if it is working in the cytoplasm or membrane or nucleus, wherever the site, it has to go there. Then also it may not be functional until it needs some associated partners. Then only it will do the function. It's not directly leads to DNA and function. It has to cross many steps. Okay. So coming to the cellular differentiation, as I told, for me and you, difference is 0.1%, 99.9% identical. Okay, but in myself or any organism, if you take different tissues like a brain tissue or muscle tissue, if you compare the DNA, that is exactly same DNA. But muscles are totally different than the nerve. You cannot just compare it. Okay, it's totally different in the morphology, difference in the functional, or difference in the gene composition. Everything is different, but although DNAs are same. That's what I want to make the point. This difference happens due to the tissue specific gene expression. Okay, so there should be some gene expression in the muscle which will not express in the nerve. Whatever the nerve specific gene, that will not express in the bone. Like that, each tissue will have certain genes that is specific to particular tissue. Okay, so these kind of cells, we started from the single cell embryo. We started from the single cell, then we will multiply, then organ formation, and we are becoming the entire human being. Okay, so we made up of individual cells, that is approximately 3.2 trillion cells. It's there, which include all the tissues in the blood cells and all the body cells. Okay, so this process, how this individual cell become the various type of tissues or cell, that process is called cellular differentiation. Okay, so that means the presence of DNA that may not be functional until the DNA message has to be translated into the protein, and that translation will not, as I told, that 20,000 genes are there in our body, so not all the 20,000 genes expressed in the particular tissue. Okay, certain genes will be expressed, certain genes will express all the tissue, that is called housekeeping genes. Certain genes expressed only the particular tissue, that is the tissue specific genes. Okay. So how this tissue specific gene expression happen? Okay. Any gene expression, it has to express or some gene has to be off. It has to receive the signal. So usually the cells will receive the signal from external or some signal, it will be internal inside the cells. Okay. Based on the cells, that the signal will get amplified through the cell signaling molecules. Or the receptor will receive the signal, then the signal will get amplified through the cell signaling molecules. Then it will go to the nucleus where the transcriptional activation complex or inactivation complex will form. That complex has the transcription factor and co-activators or co-repressor and some non-coding RNA. All the molecules will assemble in the promoter or regulatory region of particular gene. Then it will decide based on these complex, it will decide whether the particular gene has to be on or off. Okay. So based on the external signal, the signal will get amplified. Then it will alter the gene expression. This is where the tissue specific gene expression happens. And many factors involved in the tissue specific gene expression like growth factors and their receptor and cell signaling molecules and some transcription factors and some epigenetic modifiers. Okay. Apart from the genetic, there is epigenetic also is there that we will talk in another occasion. So the splicing factor and non-coding RNA. So these are the molecules involved in the tissue-specific gene expression. Among these, transcription factor, it's considered to be master regulator. Why we are considered as a master regulator? Because you take one transcription factor, put it in one cells. Okay. For example, I am taking muscle-specific transcription factor, MyoD or anything. That if I put it in the fibroblast, now the fibroblast cells can be converted to muscle. So these transcription factors are master regulator, which can turn on or off the entire lineage. Okay, that's why we call it as a master regulator, the transcription factors. That transcription factor will know which gene to be activated or which gene to be off. Okay. So coming to the altered gene expression. So all the disease, if you take all the disease, you can see that the central dogma, if any problem in the mutation in the DNA or any problem in the RNA expression or problem in the protein expression or it is altered in the different way or the functional aspect it is stopped, any scenario, it can lead to disease development. Okay, any condition, either DNA mutation also affect the protein function and any level, any change happens that leads to disease development. So deregulation or alteration of gene or its expression involved in almost all disease. Altered expression and function of various tissue-specific transcription factor also reported. The same way, tissue-specific transcription factor also getting altered. That means it's altered functionally it is altered. Altered means overactivated or underactivated or non-functional. Anything we can tell. It can be altered that leads to disease development. So we need to understand the normal regulation, what happened in the normal development. Then we can compare the disease, what is the changes. Then we can come with a strategy how to manage the disease effectively. Okay. So this is the just introduction of the gene expression and the function. So coming to the cancer, as everyone knows, that it is a very complex genetic disorder. It's not a single gene involved. Many genes are involved. That's why we are calling it as a complex genetic disease, and which is caused by uncontrolled proliferation of abnormal cells. Okay, this is one of the leading cause of death worldwide. You know, and there are 100 types or more than 100 types of cancers are there. And if it is left untreated, it leads to death. So coming to here, these are the common type of cancer. Anyway, if you see some diagnostic report, you see some name of the cancer. Sometimes we don't understand. Okay, if you know that why they are, what are the common types, then you can easily understand that cancer from the derived from the particular cells. Okay, these types are done by the cells of origin. You know, any abnormal cell in our body can become a cancer or tumor. Okay, so based on the origin, which cell it is originate, then they named this a different way, like a carcinoma. Carcinoma derived from the epithelial cells, like a breast, prostate, lung, pancreas, colon, and all. It's formed by the epithelial cells. So the cancer derived from the epithelial cells are called carcinoma, and the sarcoma is from the connective tissues. like a muscle, bone, cartilage, and a nerve. And these are the connective tissues. So any cancer derived from the connective tissues called sarcoma. And lymphoma, leukemia, generally called as a liquid cancer, and other cancers are called as a solid cancer. That means you can see that lump. Whereas in the liquid blood cancer and all, you cannot see the lump, but that increased number of cells will be there. Okay. Lymphoma and leukemia, it is in the hematopoietic cells, and some germ cell tumors, which is derived from the pluripotent, immature precursor cells tumor, and the blastoma. Blastoma also precursor cell. So you know the one cell, I told that one cell become the various tissue. Okay, that process is called, the cells will become committed to make the single cell become the muscle or nerve or bone. First, it will commit to particular lineage. One cells are committed to muscle, then it will convert to the muscle. Other cells are committed to nerve, it will convert to nerve. That is the commitment stage. Okay. So that stage still it retains the proliferative potential, but that is the progenitor for particular lineage, like a muscle progenitor. Okay. Any cancer derived from this kind of progenitor cell, that is called blastoma. Example, retinoblastoma and neuroblastoma, medulloblastoma, and many blastomas. If any names you heard with the blastoma, you think that it's a progenitor cell of particular tissue. And adenomas are glandular organs, glands. Okay, glandular organs are called adenomas. So coming to the causes, what causes the cancer? So these are the everyone knows. Okay, the genetic factor and the environmental factor, and if you are consuming tobacco and alcohol, that can lead to cancer. Some physical inactivity also can cause the cancer. Certain infection like HIV or HPV, certain virus, if it is infected, can lead to cancer. And the overall environmental exposure to various type of chemicals or radiation can cause the cancer. But if you take one by one, this is not real. For example, alcohol or tobacco consumer will get the cancer. If you take all the alcoholic getting cancer, no. If everyone is consuming the tobacco, but still certain people are getting cancer. Okay, so it's not that simple. We cannot just tell that genetic factor is altered, you will get the cancer. Tobacco you consume, you will get the cancer. No, it is not definite. Still the definite cause how single cell become the cancer, still it is not known. But what is this factor? This factor can trigger the cancer development, which can aid the cancer development. But still, this is alone is not sufficient. It is a combination of factors, various factors, which drives the cancer development. Okay. So we are seeing the cancer and cancer. What is this cancer? What is the nature of this cancer? Okay. This is coming from the famous review article about the cancer from the Hanan and Weinberg. In the year 2000, they published the review article. There they proposed the cancer. If any cells have this kind of characteristics, then we can call it as a cancer. So initially they proposed 6 hallmarks of cancer. Then recently in the 2011, later on they proposed some of the additional hallmarks. Because we are studying the cancer and we are understanding more, then the cancer is also becoming more complex. Okay. So coming to the hallmarks, one is the sustaining proliferative signals. So any cell has to grow, it has to receive the growth signal. Then it will grow. So, but cancer don't need that signal. It can make its own signal and it can survive or it can grow on based on their signal. Okay. And another thing that evading growth suppressor. When the some cells are growing abnormally, see, we are growing, I am growing. I stopped the growth here. Further, I am not going continuously growing, right? Okay. Certain stage we are stopping the growth. Okay. That is done by the growth suppressor. But this cancer can escape from the growth suppressor. Wherever the genes or whatever the molecule which try to stop the growth, but the cancer will not follow the growth suppressive event. Okay. And also another important hallmark is activation of invasion and metastasis. Cancer cell can invade to neighbor tissues. It can move to neighbor tissue or it can metastasize to distant organ. Like a cancer happened in the leg or something, it can go to the bloodstream and it can go to the brain. It can make the secondary tumors like that. So that process is called metastasis. Okay, these are the another properties of the cancer. And another thing is the replicative immortality. Any cell, if you take it, you grow in the culture, it will go certain lifespan. Okay, it will grow in the certain time. After that, it takes the senescence pathway or the cell aging process. After certain pathway, it will leads to aging process. Whereas cancer, it has the replicative immortality. Okay, it will just continuously proliferate. That is the another property of cancer. And another thing is the inducing angiogenesis. Angiogenesis is the blood vessel formation, because the cancer, if you see the cancer is there, the blood vessels are going around. If some tissues are in the middle of the cancer, it will not get any nutrient. So for that, cancer will make its own blood vessels. That is process is called angiogenesis, to get the nutrients from the blood. Okay, that is the angiogenic process, also another hallmark. And resisting cell death. If you are trying to kill the cells, and these cells are very resistant to the cell death signaling. Okay, so these are the 6 hallmarks of cancer. And another emerging hallmarks like deregulated cellular energetics. For example, for us, the cell energy will take by the any process, any glycolytic pathway or the lipid pathway or any metabolic pathway, it will take the energy. Okay, but cancer cells will take the alternative energy. It will not take the conventional energy how the normal cells are taking. Okay, that is a deregulated energy pathway. And also the immune system. Immune system plays a double role. Okay, opposite role. So it can avoid the immune system. Immune cells, we can recognize the cancer, it can kill it, but the cancer will escape it. One thing. Another thing, that some immune system, some immune response can promote also cancer. Okay, so that's why telling that immune system has the 2 opposite role. It can block the cancer also, it can promote the cancer also. That's what we understood. And the genomic instability. If you take normal, there is a mutation rate is less, but in the cancer, that mutation rate is more, because the DNA repair enzyme or DNA replicating enzymes are defective. So it can make the defective DNA. So that's why the DNA or the genome present in the cancers are highly unstable. Okay, that is the highly that is the unstable genome that leads to mutation or alteration. So coming to the cancer gene, there are 2 sets of genes. You can group the cancer genes into 2 sets. One is the oncogenes and another is the tumor suppressor gene. So you know the oncogenes, any genes which promote the normal growth and development. That time, if you promote the normal and growth development, it is called proto-oncogene. When it's getting mutated or altered, which can become the oncogene, then continuously promote the cancer development. Okay, so proto-oncogenes, nothing but any genes which promote the growth and the development and the proliferation of any cells. For example, make PDGA grow, all the growth factors and their receptor will come under the oncogene. Tumor suppressor gene is certain genes which can block the tumor development. Okay, for example, any genes which control the cell division, cell growth, proliferation, and some genes which can repair the DNA mistake. If any DNA is damaged, the DNA repair genes will be there, will go, and which will correct the DNA mistake. So these DNA mistakes genes are mistake error repair genes are called tumor suppressor gene. And some genes induce the cell death like apoptosis. Those genes also called tumor suppressor gene. So any gene which can activate the, which can promote the cancer is called oncogene. Which can suppress the cancer or the growth and development is called tumor suppressor gene. For example, this oncogene, any mutation, you know that we have the 2 allele. Any gene, if you take it, that has the 2 copy. Okay, any chromosome will have the another copy, right? So alleles, we have the 2 alleles. For the oncogene, any one allele is inactivated or altered, which can drive the cancer. Whereas the tumor suppressor gene, both allele has to be inactivated. Both has to be off. Okay, so if I explain the other way, for example, you are riding the car. Okay, if any problem in your accelerator, your car is going to undergo the accident. Okay, it can go to uncontrollably. You cannot control the car if the accelerator is gone. That means one change is enough. For oncogene, one change is enough. If you consider the tumor suppressor gene, it's like a brake. You have the brake in the front and you have the brake in the rear. Okay, so if front brake is failure, still you can control the car with the rear brake. Like that, tumor suppressor has the two copies. One copy is inactivated, still the other copy will suppress the tumor. Whereas the oncogene, one change is enough to create the cancer. That is the thing. Okay. So when the single cell will develop the tumor. Okay. There are two events. One event is called tumor initiator or cancer initiator. Another event is the cancer promoter. So if any initiator happens, there will not be any tumor. Initiator followed by the promoter then leads to tumor. That promoter may be immediate. Initiator followed by promoter immediate, or the promoting event may be after 20 years, after 10 years also it can. But initiator followed by the promoter leads to tumor. Only promoter, there will not be tumor. All the many promoters followed by initiator, still it will not form the tumor. So you need the initiating event, cancer initiating event, followed by various promoting event, then leads to cancer. Okay. What is these events? Initiator event. That's what we saw in the cause of the cancer. It can be DNA damage, or it can be chemical exposure. It can be anything. So initiator event can be anything, or genetic factor. Promoter event also can be similar thing, but any initiation event followed by the promoting event leads to cancer. If it is altered, it will not develop the cancer. Okay. So coming to the cancer, cancer is highly heterogeneous in nature. Heterogeneous means what? Different type of cancer behave differently. Brain tumor will behave differently. Bladder cancer will behave differently. Lung cancer will behave differently. Not all will behave the same way. Their growth rate is different. If you treat it with a one drug with the brain tumor, or another drug with a, this treatment response, how it responds to drug also varied. Therefore, necessary for the type specific treatment. For example, normal cell also will be there in the tumor. And some due to the increase the time, there will be a lot of abnormal cell or different type of cells will be there. So it's a heterogeneous population. Cancer is not a homogeneous population. But coming to the therapy, what is the cancer treatments are available? Surgery is the effective form of treatment. Wherever you are seeing the lump, you can remove it. And also chemotherapy. Chemotherapy, what they are giving the chemotherapy drug? Some DNA damaging agents, some antimetabolite, anti-tumor antibody, some topoisomerase, some enzyme inhibitor, some mitotic cell proliferation inhibitor. Okay, these are the chemotherapy drug. And radiation therapy is available. What they will do? They will pass the radiation to kill the cancer cells. Okay, there are another emerging therapy, immunotherapy, hormone therapy, some of the targeted therapy also emerging. So as I told, the cancers are heterogeneous, but the therapies are non-specific. For example, DNA damaging. It will not know to damage. For example, DNA damaging agents, if you take cisplatin, cisplatin will create the DNA damage in the normal cell also, tumor cell also. Okay. As I told, the cancers are heterogeneous, but therapies are common. That's why the therapies are failing. And another thing that what caused the cancer, we saw the chemicals. Okay, chemicals, how it will cause the cancer? Chemical will leads to DNA damage and it will create the mutation that leads to cancer development. But this chemotherapeutic drug also same thing. This chemotherapeutic drug also creating the DNA damage. So whatever we are thinking it's a cause of cancer, the same thing we are giving as a therapeutic. Okay, you know that radiation exposure causes the cancer, right? But the treatments are radiation treatment. What will happen radiation? If you are getting radiation exposure, you will get your DNA damage. If the DNA damage is there, your cells will become abnormal. Then once in the point, it will develop the uncontrolled growth. Okay. So this therapy, whatever the available therapy, see, these are the managing therapy. That's why that there is no definitive cure. Because of this therapy, certain cancers are improved the survival. But the thing that is certain patient, it's not completely definitive cure for all the patients. Okay. Yes, I agree that because of this therapy, there is a little bit improvement. The survival may be 5 years to 10 years, 10 years to 15 years. But the complete cure is not available because cancers are heterogeneous population, but our therapies are homogeneous, single kind of therapy. Okay. So therefore, the targeted therapies are required. What is targeted therapy? Targeted therapies are drugs or any substance that can block the growth and spread of cancer by interfering with the specific molecule that are involved in the growth, progression, and spread. Any hallmark, you take that, you can target to angiogenesis, you can target to migration, you can target to invasion, like that. You can target to any hallmarks of the cancer, you can target the drug. That is called targeted therapeutics. So what is the benefit of targeted therapeutic? It can prevent the cancer cell growth. It will not touch the normal cells. And it can interfere with the tumor angiogenesis, which can promote the death only the cancer cell, which can stimulate our immune system to target the cancer also. And you can deliver the toxic drug. Whatever the chemotherapy we told that it's non-specific and it's toxic, that drug you can deliver only to the cancer cell, not the other cell also. That is possible by the targeted therapy. Overall, it's considered to be less side effect. There are a lot of targeted therapy drugs are developed already and it is approved. Like imatinib, gefitinib, sunitinib, bortezomib. These are targeting the different molecule. For imatinib and all, it's targeting tyrosine kinase receptor. And some growth factor and the receptor growth factor function it is inhibiting. But still, this, although it is there, it is not targeted. As I told, any signal cells receive, it will get amplified. So they are blocking in that receiving place. But the amplification is the various molecules are involved. Okay. So this also not proper target, because here also we are getting a lot of side effects. Like a EGF not only involved in the cancer, it is involved in the skin development, normal proliferation. So if it is blocked the EGF, also you reduce the cancer and you reduce the skin proliferation. Okay. So like that, whatever the targeted therapy, it is targeted, but it's not the right target. So we need to identify the right target. What is the weak point? Like here in this image, it's showing the Duryodhana's weak point. Okay. So if you identify the weak points of the cancer, like a specific target, then the target will be useful for the cancer treatment. Okay. So we need to identify the right target. And coming to the differentiation and cancer, as I told that various tissues are forming the cellular differentiation process. So before going to that, and the cells will get committed first. Progenitor cell will get committed first. Once it's committed, then it will undergo the differentiation process to become the various cells. Okay. So during this committed stage, if any problem happen, it will not complete the tissue formation. Rather, it will just continuously grow, develop as a cancer or any disease. And some cells are completed differentiation. Okay. For muscle or completed differentiation. Once it's completed differentiation, the proliferation will go down. As I told you, you can see that during the differentiation, that proliferation will go down. The same time, differentiation capacity will increase. Okay, it's just opposite. So once it's differentiated completely, there will not be any proliferation. It's a terminally differentiated. Somehow this cells gains a proliferative advance, that will become the cancer. So during the differentiation, any block which can lead to cancer, or completed differentiation, somehow it escapes from the differentiation thing, it starts proliferation also leads to cancer development. Okay. So any deregulation in the cellular differentiation leads to cancer development. And this cancer, there are a lot of transcription factor, tissue specific or the transcription factor involved in the various process of the differentiation. Those transcription factor getting altered in the cancer. So coming to our research focus, development of safe targeted therapeutics. So for that, as I told, the transcription factors are master regulator. Transcription factor involved in the process of getting the message from the DNA to RNA, and the transcription bind to promoter or enhancer region, which can control or which can on the gene or off the gene. Okay. Tissue specific gene expression factor allow the unique expression. The tissue specific genes are expressed because of the tissue specific transcription factor. So this transcription factor will play a lot of role in the development. When we are starting from the single cell, various tissue formation, this will going up. Once the development is completed, once the muscle is formed, once the nerve is formed, once the bone is formed, this expression will be off. Or it will go to particular tissues, not all the tissues. Okay. What happens in the cancer? Certain kind of tissue specific transcription factor re-expressed. Okay. So that may leads to cancer. So the reactivation of the silenced gene leads to disease development. So we can target, pharmacologically targeting this factor. It's a key challenge. It's very difficult to target the transcription factor. But if you are targeting transcription factor, that is a better. Okay. So coming to our work, and we are using the transcription factor PAX3, that is the paired domain transcription factor involved in the developmental pathway of neuron, melanocytes, muscle development and all. So overexpression of this gene or re-expression of this gene reported in the many cancer, like a glioma, neuroblastoma, melanoma, rhabdomyosarcoma, many cancer it's reported. But the existing literature shows that different roles, how it is involved in the cancer, it's different in role. However, the exact function also not known. But in our work, we want to study the function, how it is plays a role in the cancer development, then whether we can target this molecule for the cancer specific target or not. Okay. So coming to this figure A, we are having the neuroblastoma, that is the tumor cell derived from the neuroblastoma tumor. Both human cell and the mouse cell we have it. First, we are trying to induce the differentiation. Okay. When we induce the differentiation, which we can convert the cell into differentiated cell, what will happen? How the PAX3 get altered. Okay. So you can see that the GM and DM. GM is the growth condition. DM is the differentiation condition. So we are culturing the cells in the differentiation condition. You can see that it is making like a neuron-like projections, like a lot of projections that the dendrites. Dendritic projection will get increased. Single cell will become more dendrites and it will make the connection also. So that is the data is quantified here. Okay. So the cells are going the morphological differentiation. When we are culturing this neuroblastoma cell, it will mimic like a neuron-like cell. Okay. So here what will happen, whether the neuron, since this is considered as a differentiation, what happens the neuron specific genes, neuronal genes. So we look at the NF68, synaptophysin, NUN, beta-3 tubulin. These are the neuronal specific gene in the figure C. You can see there is no alteration in the growth and DM differentiation condition. Okay. Both mouse cell, rather some cell like a synaptophysin, it's reduced in the mouse cells when culturing the differentiation condition. So that means when we are pushing the tumor cell into differentiation, it mimic or it behave like a differentiated cell, but in general, the genes are not upregulated. But morphologically, it is showing the differentiation. Okay. So in this condition, what happens to PAX3? PAX3 already known that it is involved in the neuronal development, neuronal differentiation. What happens to the PAX3 genes? So we looked for the PAX3 gene expression. So we did not find any change. PAX3s are just like that expressing. There is no change in the growth condition and differentiation condition. Okay. And we looked at some PAX3 target, although we can see the PAX3 is expressed. What happens to the target, whether it is active or not? So we are using the figure B. You can see that NeuroG, NCAM and all PAX3 target. So there is no difference in the PAX3 target also. So that means the PAX3 is there. When we are converting the cells, pushing the cells to differentiation, PAX3 is not getting altered and it is also not showing any functional alteration. For the PAX3 to induce the neuronal differentiation, excuse me. PAX3 to induce the neuronal differentiation, it has to be functionally active. But here, the functional is not getting altered. And in addition, the PAX3 has the 8 different isoforms. You can see in the figure C, PAX3 A, B, C, D, E, G, H, I, 8 different isoforms, which has the different domains of PAX3. Okay. PAX3A has only DNA. Any transcription factor will have 2 domains. One will be DNA binding domain, another will be transactivation domain. So DNA binding domain, it will just go to the bind the DNA and activate the gene. The activation, gene activation or gene repression happen by the transactivation domain. So DNA binding domain will be there, transactivation domain will be there for the any transcription factor like that PAX3. It has the parent domain and homeo domains are DNA binding domain. TA transactivation domains are there. You can see that various isoform is there. Some isoform don't have the transactivation domain. Then we see what kind of isoform expressed in the neuroblastoma cells, where in the figure C, you can see that RT-PCR data, you can see that human, it expresses all the isoform, both inactive isoform and active isoform also. Whereas in the muscle, only inactive isoform PAX3A. You can see that PAX3A don't have the transactivation domain. So that means the PAX3 is present, it is inactivated because it exists, majority exists as a inactive isoform and the active isoform is not there. Okay, that's why it is not activating the neuronal specific target gene. So you can see that mouse also, when you are culturing the growth to differentiation also, there is a not change in the isoform expression except certain active form. Whatever the resting active form is there, that also going reduced in the differentiation condition. So that means the PAX3 has to be activated to induce the differentiation. Okay. So coming to the activation. So to confirm it, if you reintroduce the PAX3 in these cells, functional PAX3 in these cells, what will happen? Whether it can reduce the tumor or no, or which can induce the differentiation or no. So for example, for this, we are introducing the PAX3. You can see that mouse control and the PAX3. That means we can add the functional PAX3 into these cells. Okay, we are transferring the functional PAX3 into the both the mouse and human cell. This is the, you can see that we increase the PAX3. Then we look the tumorigenic property like a growth property, colony formation property, and we saw, you can see that and the black histogram and the white histogram is a control. The black is reduced than the white. Okay, that means that when we introduce the PAX3, it is stopping the growth, cancer cell growth. And also the another property is the colony formation. When you grow the cells, it will make the individual colonies. Okay, that colony formation ability also it's reducing when we are introducing the functional PAX3. That means if your functional PAX3 is there, it can inhibit the growth and also we can induce the differentiation. That is the hypothesis. To activate the PAX3, so we need some activator. Okay, for that we screen the activator, potential PAX3 activator. So you need that, you cannot give the PAX3 gene therapy, that will go as a gene therapy. So gene therapy has a lot of complications, a lot of side effect and a lot of unverified roles are there. We don't know what is the long-term effect. So we cannot just introduce the PAX3 into the cells. Okay, to overcome that, we are trying to identify some chemicals or from drugs which can activate the PAX3. Okay, so for that we use the chemical library, like a book library. We have the various chemical library. So we screen the 3000 compounds, various molecule. It's a known molecule. Then we develop the. Before going there, I will tell the system. So for to develop the activator, as I told that PAX3s are inactive in these cells, functionally inactive. If you introduce the functional PAX3, it can block the tumor growth. Okay. So for the activator, we generated the system. Okay, cell-based readout system. How we introduce the system? You can see that 6xPR, that is the PAX3 binding region. As I told that any transcription factor will bind to the DNA. So we know that where the PAX3 will bind, that DNA part we took it, that is called PAX responsive sequence, that we attached with the luciferase. That is a reporter gene. We construct the gene. Whenever the PAX3 is there, it will bind to the PAX responsive sequence. It activates the luciferase. So that means indirectly you can measure the activity of PAX3 by the luciferase activity. Okay. So the PAX3 is there, it will bind to the PAX3 DNA, that is the PRS. Because we attach the PRS with the luciferase. So whenever the PAX3 is there, active, it will bind to there and it will activate the luciferase. So in the figure A, you can see that IMR only cells, the cells introduced with the luciferase. So you can see that luciferase activity is increased. Okay. So that luciferase activity is further enhanced when we introduce the PAX3. Okay. You can see in the right side, in the bottom figure D. Okay. Figure B, you can see that we are introducing the PAX3 into the cells. Okay. In the Western blot shows the less PAX3 in the 293FT. And the 293FT PAX3, we exogenously introduce the PAX3. Okay. So you can see the luciferase activity in the when you are control. less luciferase activity. When you introduce the PAX3, more luciferase activity. When you remove the PAX3, that is SHRNA mediated. When you remove the PAX3, the luciferase activity is going down. So that means whatever the luciferase activity we are measuring, that's due to PAX3 activation. Okay. So we developed this system. Then we screen, we apply the various chemical molecule. Then we will look which molecule can activate the PAX3. That's what we did it here. We applied the 3280 chemical compounds. Then we got some kits. Then we passed to dose test. Then we pick up some 16 molecule. Then we use the PAX3 specific gene activation. There we found the 4 molecule. You can see in the figure C, the PAX-A1, A4, A8, and A10. Okay, these are the molecule can activate the PAX3. Okay, so now we have some molecule which can activate the PAX3. Already we have the result that when we introduce the PAX3, it's blocking the growth. Then whether these activator can block the growth or no. So we studied in the various cell lines. Okay, so we use the various cell line with the different PAX3 activator. So in this, among the 4 activator, PAX-A1, you can see in the IMR cells, PAX-A1 and PAX-8, both is reducing the tumor growth. And in the neurotube also, the PAX-A1, it's reducing the tumor growth. Then we analyze the various tumorigenic property like a colony formation. You can see in the image B, PAX-A1, you can see that it's reducing the colony formation. And also PAX-A8, PAX-4, PAX-10 did not affect the growth. Maybe we need to adjust the concentration or something. But PAX-A1 and A8 can reduce the growth. Okay. So we identified certain molecule which can activate the PAX3 that also blocks the cancer cell proliferation and the colony formation. And we also tested with the migration and other property also. Still, these molecules can have the inhibitory role in the migration and invasion also. Okay. So overall, what happens in the neuroblastoma? In the neuroblastoma, the PAX3 exists in the inactive isoform. The PAX3s are transcriptionally inactive. Okay. Because of the transcriptionally inactive, it cannot induce the differentiation. Rather, it develops the cancer. When we ectopically express PAX3 into these cells, which can prevent the growth and the colony formation. Okay. So we develop the PAX3 activator that also shows the reduce the growth, tumor growth, and the other properties also reduce. Okay. So that means in the overall, this PAX3 activator can be developed as a anti-cancer therapeutics for the cancer where the PAX3 is involved. Okay. So now I'm moving to the another cancer that is a different molecule in the here that is called Rhabdomyosarcoma. So far we saw the neuroblastoma that is derived from the neuroblastoma. Blastoma, I told that it's a progenitor cell. It's a neuroblastoma means neuronal cell progenitor it forms. Okay. Now coming to the Rhabdomyosarcoma, it's a common soft tissue sarcoma in children. Okay. There are 2 major types: alveolar rhabdomyosarcoma, embryonal rhabdomyosarcoma. Alveolar counterpart is a highly aggressive tumor than the embryonal. Okay. These cells are defective in converting to the muscle differentiation. Okay. So they also often shows the defective muscle differentiation. These are the tumors of muscle origin. Okay. It will not convert to the muscle. Although it is expressing the transcription factor like MyoD, myogenin, but still it cannot convert to muscle. It is developing as a tumor. Okay. So coming to the muscle differentiation, like a neuronal differentiation, any differentiation, there will be progenitor cell that will undergo the committed myoblast stage, myoblast proliferation. Then the myoblast will fuse to give the myotubes. The myotube leads to muscle formation. This is the normal muscle differentiation. During this various process, various tissue specific transcription factor involved. For example, the PAX7 or PAX3 involved in the progenitor stage. Once it's going to the myoblastic stage, the expression will go down. Then the Miphail, another gene, Miphail transcription factor will take role and it maintains the myoblast stage. Once it's undergoing differentiation, the Miphail will also go down. Then the same time the MyoD, another transcription factor will go up. It takes the other role. Then once the role is over, it will go off. Then the another gene, myogenin or MRF will take the role. then that will complete the differentiation. During this differentiation, various gene, various transcription factor will go up and down, and it regulates the particular stage. Okay. So the normal muscle differentiations are controlled by various myogenic regulatory factors like a MyoD, myogenin, Miphail, MRF4, and deregulation of these transcription factors or the expression leads to muscle cancer, that is a rhabdomyosarcoma. Okay, so here we are targeting the transcription factor Miphail. Okay, so Miphail is the gene belongs to MRF family, myosin regulatory factor family. And like any other transcription factor, if you put Miphail into the fibroblast, now the fibroblast can be converted to muscle. Okay, so the Miphail involved in the commitment stage, not in the differentiated stage. Okay, the role of Miphail in the muscle cancer development is not explored. So first we want to understand what is the role of Miphail. First we see what is the role of Miphail in the normal differentiation. So we use the C2C12. It's a mouse myoblast cell. If you grow the myoblast, we can convert the myoblast into myotubes. Okay, you can see the figure B. You can see that long cells. That is a myotubes, multinucleated myotubes. So you can see in that figure, myoblast, the individual cells, multinucleated myotube, individual cells will fuse and gives the myotubes like structures. Okay. So during this myotube formation, the Miphail gene, you can see in the figure A, Western blot Miphail, top panel, the Miphail gene will go down. The same time, MyoD will go up. Okay, the Miphail involved in the proliferation. During the differentiation, Miphail will go down. The same time, MyoD will go up and undergoes the differentiation. You can see that MyoD is up, and the myosin heavy chain, that is a terminally differentiated cell marker, that is going up. Early gene is a myogenin is going up. So during the differentiation, early gene, late genes are going up, and whichever is involved in the proliferation, like a Miphail, it is going down. The same thing is in the RT-qPCR also here. So Miphail is going down, other genes MyoD, myogenin, myosin heavy chain, and another CDK1A, that is a P21 inhibitor. When the cells are growing differentiation, the growth will stop. Then it will go to differentiation. So this is the growth arrest gene. So growth arrest gene is upregulated. During the differentiation, growth arrest gene is upregulated, and which will block the growth. Then the cell will convert it to muscle. Okay. So during this process, Miphail gene is going down and the MyoD is going up for the normal differentiation. Then we look in the tumor cell. What is the expression of this factor, like a Miphail or MyoD? So you can see that we use the 4 different cell line of Rhabdomyosarcoma, where we can see that Miphail expressed in the U20325, U210415. So these 2 cells, Miphail is expressed. Okay, and MyoD also express the various level. But if you carefully note, whenever you are seeing Miphail, you are seeing less MyoD. Whenever seeing more MyoD, like a U20325 cells, you can see the less Miphail. So the MyoD and the Miphail mutually exclusive. When the MyoD is there, you can see in the previous also, C2C differentiation, normal differentiation, the Miphail will go down, the same time MyoD is going up. Okay. So when we are pushing the tumor cells to differentiation, what will happen? Okay, this is the normal cell differentiation. Miphail is going down, MyoD is going up. When the tumor cells are differentiation, what's happening? So when we are pushing the cells to differentiation, tumor cells to differentiation, we saw the Miphail is increased. In the normal cell differentiation, the Miphail has to go down, MyoD has to go up. Whereas in the tumor cell, if you push the cells to differentiation, rather the Miphail is to go down, it is increasing. At the same time, if the Miphail is increasing, MyoD will get decreased. MyoD is necessary for the differentiation. So if the MyoD is blocking, Miphail is increasing, which blocks the MyoD, which blocks the differentiation. Okay, this is the hypothesis, whether the Miphail is negatively affecting the differentiation or not. So for that, we are taking the cell, we exogenously introduce the Miphail into the cells. Okay. Then we look for the differentiation specific gene. You can see the figure B. You can see the myotube formation in the normal. On the bottom figure, you can see that when we introduce the Miphail here, which is blocking the differentiation. Okay, that means the cancer, when we are pushing the cancer cell to differentiation, the Miphail is going up. So that is the responsible for the defective differentiation in the tumor. Okay, that we showed in the normal myoblast. When we introduce the Miphail, it is blocking the differentiation. Okay, and that is quantified in the figure C also. So coming to the conclusion, normal myoblast, when undergoing the muscle formation, the Miphail expression will go down. At the same time, MyoD expression will go up. Whereas in the cancer, instead of going, the Miphail is going down, the Miphail is going up. Okay. Then we don't know how it is developing the Rhabdomyosarcoma, ARMS, whether it's a myoblast differentiation is blocked or the terminal differentiation escaped some cells, develop the tumor. We don't know what is that scenario. But any scenario, if we try to push the cells to differentiation, the Miphail is going up and it's blocking the differentiation. Okay. So for that, we are hypothesizing that if we develop the Miphail inhibitor, which can block some Miphail function, so we can develop the targeted therapeutics for the rhabdomyosarcoma. So for example, already we saw that if we target the activator of PAX3 can develop as a targeted for the neuroblastoma, here the inhibitor for the Miphail can have the potential for the neuroblastoma targeted therapeutic. This and all in the research phase only. And we are currently in our lab, we are doing some other work also apart from these things. And we are evaluating muscle neuron specific other transcription factor. What are the various transcription factor, whether those transcription factor can be targeted or no. And we are also focused on the development of splicing factor targeted therapeutics. As I told, the tissue specific transcription factors are done by the tissue specific gene expression carried out by the transcription factors, and also splicing factors also involved in the tissue specific splicing event. So we are developing whether we can target the splicing factor also, and also we are exploring some non-coding RNA. Okay, so we know that whatever the RNA produced, as I told, the DNA, the message is converted to RNA. So whatever RNA is produced in the cell, 98% are junk, which is not code for anything. Only 2% code for the protein, whatever functional. The 98% are non-functional. That is called non-coding. Okay, there are recent reports. There are many reports sourced. Although it is expressed, this is a functional only. Okay, so what is the function? Still, people are trying to understand the function. Okay, in our lab also, we are trying to understand the role of non-coding RNA in the normal differentiation and its alteration in the disease also. Okay, so apart from the transcription factor, we are also targeting the splicing factor and some non-coding RNA also. Okay, so that's what I'm concluding my session today. So I would like to acknowledge our director, our administrator, director, registrar, deans. And also, I would like to thank my students. They are the PhD scholars. Abhinaya Selvi, she is ICMR. She did a lot of work in the muscle differentiation. And Naren Kumar, he worked in the neuroblastoma and the PAX3. And Soundarya Ladas, he is working on the brain tumors and role of the PAX3. Okay. I would like to thank my international collaborator, Dr. Maul and Dr. Gabellini, and also the national level collaborator, Dr. Murugesh Pondian Egambaram from Bardiyar University. And the Dr. Prabhraj, he is a neurosurgeon. He used to give our brain tumor tissue, and the Dr. Nandis, he is a neuropathologist. So I would like to thank everyone, and also the funding agency, SERB, SERB, and also the NIMANS. So I would like to thank all the support, and thanks for the listening. If any query, I'm happy to address your queries.
Thank you, sir. The session is now open for question and answers. So participants, please type your questions in the chat box, and sir will be able to answer them.
Students, you can even unmute yourself and ask question.
Madam, students won't be able to unmute actually. We are in a Zoom.
Okay, Chandan. Okay.
So we have one question from one of our MSc students. He says, which programs do you use to select a few of those 3,000 compounds for PAX3 activation?
Okay, there is no program. These are the libraries. These are the small molecule chemical libraries are available. different library has the different format and different number of molecules. Here we use the two different library. One is from the spectrum, that is a LOPAC library from the Sigma. Another is that spectrum library from the MDS, Microsoft Discovery System. Two different companies, they have the different library. One library has 1250 compounds, another library has the 1000 compounds. So we combine the 3000, we made. And not only this library, we have the bigger library. This is the preliminary screening we did with a small library. And we also screened the 50,000 compounds from the Cambridge library. Okay, there also we identified that is unknown compound. First, we want to try with the known functional compound. If our strategy is working, then we are developing the drug from the unknown library. Okay, there we can pick our molecules. There is no program we can choose the library molecule. Libraries are existent, which has the known chemicals or unknown chemicals, which functions are known or not known. Different molecule libraries are available. So I can see in the chart box which program. Okay, yeah, that I answered it. How do you check the effectiveness, sir? So the effectiveness, as I told, that we generated the system where we can measure the PAX3 activation, PAX3 function. So if you introduce the drug, whether the drug is activating PAX3 or not, we have the system, that is a cell-based readout system. Based on the system, we can tell that this molecule is PAX3 activator or not. So further we are confirming with the, when we remove the PAX3 or increase the PAX3, then we can increase the activity or decrease the activity. So same way, and we have the different filtration system. Okay, one system is that to filter the general transcription activator. So the 3000 molecule is getting activated. So there will be some PAX3 specific activator. There will be general transcription activator. So we are filtering the general transcription activator with a different system. Then we are picking only the PAX3 activator. Okay, and also we are filtering with the toxic one. Our goal is to identify the molecule which is non-toxic. Okay, non-toxic or it's a less side effect. The side effects comes because of the toxicity. So we are doing the toxicity screening. We are removing the molecule which is general toxic, which is targeted only to the tumor cell. We are screening. Okay, like that we are seeing the effectiveness of this molecule also, and in terms of the PAX3 activity or the modulating the transcription factor also, and we are looking in the effectiveness or efficacy in the tumor various tumor properties also. Thank you.
Students, if you have any more questions, you can either type it in the chat box or you can ask in the question and answer section.
So what are the in silico tools available in this study? So we are not doing in our study, we are not doing any in silico, but in silico tool to analyze the, for example, here the PAX3. So we are doing the lot of NCBI has a lot of tool to identify the PAX3 targeted DNA region. Okay, the PAX3 responsive sequence has a certain motif, like a T, T, C, like that. CA, the Miphail and all will recognize CA, N, and TG. Any sequence with has the CA, then any nucleotide, then the TG, that can be recognized by the E-box proteins. Like that, that kind of a bioinformatics tool is available in the NCBI, where we can see the sequence and we can analyze the sequence, what all the transcription factors are binding. Okay, there. But for this here, the screening and all, we are not doing any in vitro analysis or any in silico analysis. And once we found to know the drug action and the drug solubility, bioavailability, there we will use the in silico screening to look the property of the unknown molecules, unknown chemical molecules or druggable molecules.
Thank you, sir. Students, if there are no more questions, we would end the question answer session. I would now like to invite.
One minute, Bhargavi. Yaran has some question. Let him ask.
Yeah, he answered that Yaran question. His question is that which program do you use selecting the 3000 compounds for the PAX3 activation? Okay, the thesis project. Yes, you can do it because our lab here, the space are very limited. So we are not taking more students, but yearly I will take the 3 candidates for the project. So this is no specific. Whomever comes first, then I will offer the project positions. And anyway, the 3 months to 6 months, different college, different university will have the different time period. So usually I will take the students 3 people at a time. So depends on the what time your project starts. But most of the college starts from December and January to March or April, the last semester. So that time people will come. So for me, no problem. So to come here, Nimans has the website training.nimans.ac.in. So you can go, then you apply through formally, then I will get it will get accepted. Okay, it will has to go through HOD and different internal things are there, but that's simple. So yearly I will take the 3 candidates for the summer, 3 candidates for the project also. Okay, because of the space limitation, I'm restricting with the 3 candidates, any 3 candidates. So whomever is contacting me first or applying the first, then I need to take the decision.
Yes, Bhargavi, you can proceed.
Thank you, ma'am. Thank you, sir. So I believe we have one more question. Oh, no, that's just him. Thank you. Students, if you have any more questions related to sir's topic today, you can ask it in the chat box or type it in the question answer section. Otherwise, we will move on to the vote of thanks. I would like to invite Professor Cynthia, Professor, Department of Genetics, to deliver the vote of thanks.
Alan James, a British philosophical writer known for his inspirational books, quoted, "No duty is more urgent than that of returning thanks." On that note, I would like to extend my heartfelt gratitude to Dr. Madhivanan Jyoti, Associate Professor, Department of Genetics, NIMANS, for sharing his valuable insights and detailed, clear explanation of transcriptional factors of PAX3 and MYF5 on cancer treatment. Thank you, sir, for taking your time from your busy schedule and being with us today. I would like to thank in absentia our Honorable Chairman, Dr. T. Somshekhar, our beloved Secretary, Mrs. Bharati, and a dear Principal, Dr. Srinidhi K. Parasarathi. I would like to also thank our IT team for their effort for the smooth conduct of the session. I extend my sincere thanks to Dr. Vanita G. Ramesh, HOD, Department of Genetics, for taking initiative in organizing this event. My gratitude extends to Professor Shubha Badran and Professor Bhargavi R. for their support and effort in conducting this session. Last but not least, I owe a big thank you to all our beloved students for their active participation. Thank you one and all.
Thank you so much, sir, for sharing your research findings with us. It was very much informative. So many mysterious things you have clarified with your presentation. It was so clear. Thank you so much for accepting our invitation and participating.